The mitochondrial electron transport chain is the first module within the oxidative phosphorylation system. It is sometimes abbreviated as ATC or called respiratory chain, and these are more or less interchangeable terms. For simplicity, we will refer to the mitochondrial electron transport chain as respiratory chain in the next part of this lecture. The respiratory chain plays two important roles. First, it oxidizes the initial electron carriers produced by the catabolic process we discussed in previous lecture on atmospheric oxygens, thus producing water and releasing vast amount of chemical energy. And second, it uses this chemical energy to pump protons across the inner mitochondrial membrane, generating the electrochemical gradient, and is then used to produce ATP. So to understand the function of electron transport chain, is important to answer these two questions, namely how it can oxidize this initial reduced electron transport carrier on atmospheric oxygen, and how it can couple this oxidation to the proton pumping. To understand this, one has to bring physical context, namely to understand what is the physical composition of the respiratory chain. In a coarse-grained manner, the respiratory chain could be brought into 6 major units. 4 of these units are large multiprotein complexes embedded in the inner mitochondrial membrane, which are referred as to complex 1, complex 2, complex 3, and complex 4. And 2 others are respectively small molecule, membrane-soluble molecule, which is called ubiquinone, and the small soluble protein, cytochrome C, which is present in the intermembrane space between inner mitochondrial membrane and outer mitochondrial membrane. Each unit within respiratory chain represents a redox factor with different affinity for electrons. For example, complex 1 has only slightly higher affinity for electrons than the initial electron carrier produced by catabolic process NADH. So this way, complex 1 can oxidize NADH, oxidize back into oxidized form, and get reduced. And because of that, it is also referred as to NADH dehydrogenase complex. In turn, complex 1 has somewhat lower affinity for electrons than this membrane-soluble electron transport carrier molecule called ubiquinone. And because of that, ubiquinone can oxidize reduced form of complex 1 back into oxidized state and get reduced to ubiquinol. And ubiquinol in turn has lower affinity for electrons than complex 3. This way, complex 3 can oxidize back ubiquinol and get triggered to a reduced form. In turn, the complex 3 has lower affinity for electrons than cytochrome C. So this way, reduced form of complex 3 can be oxidized back to oxidized state, and the reduced form of cytochrome C has low affinity for electrons than complex 4. So it can be oxidized back to oxidized state by complex 4, and this triggers the complex 4 reduction. And the reduced form of complex 4 can be turned back into oxidized form by oxidizing it by atmospheric oxygen, which is a terminal electron acceptor, which gives rise to water. This answers our first question: how the respiratory chain can oxidize initial electron carriers on atmospheric oxygen. And this occurs by a gradual transfer of electrons from the low electron affinity reduced electron transport carriers through the chain of electron affinity gradients produced by complexes within electron transport chains that finally end up on the highest electron affinity compound, atmospheric oxygen, which works as terminal electron acceptor. So when this generates a flux of electrons through this electron transport chain, the second question is how this flux could be used by respiratory chain to uh, uh, produce the proton gradient across the inner mitochondrial membrane. And to understand this, one has to consider that the transfer of electrons from the low electron affinity compounds to the high electron affinity compounds would release free energy. For example, the transfer of electrons by oxidizing the reduced form of complex 1 by ubiquinone would release free energy. And this free energy is harnessed by the large protein complexes within the respiratory chain, complex 1, complex 3, and complex 4, to power the conformational changes within these complexes. And these conformational changes in turn allows the cycles, the conformational cycles within complexes such that they could accept protons on the inner side of the inner mitochondrial membrane within the mitochondrial matrix and release them to the outside of the inner mitochondrial membrane. And these cycles, which are generated by flipping these multiple components between oxidized and reduced state during the gradual transfer of electrons, is the force that powers the generation of proton gradients by the respiratory chain. So when one could bring an analogy between the respiratory chain and electric pumps. In the case of the respiratory chain, the electric force, the electric potential is generated by the difference in electron affinity between initial electron carriers, NADH, FADH2, etc., and the terminal electron acceptor. And this change of potential electric tension is used to pump the flux of electrons through the respiratory chain, and this electron flux could be considered similar to electric current as in a pump that is used to pump material. And in the case of electron transport chain, these materials are single atoms protons single atoms protons that are pumped across the inner mitochondrial membrane. So altogether, the function of respiratory chain could be compared to function of regular pumps, with only difference that instead of normal matter like water or air, this pumps protons, and it has a very minute size of the molecular scale. As in the case of every electric device, the respiratory chain must be equipped with the conducting substance that allows the flux of electrons, electric current to occur through the respiratory chain. And in this case, it is the function of electron conducting substance is accomplished by multiple electron carrier compounds which are present within the spread around within various elements of the respiratory chain. Some of these we already touched upon in the previous slides, but altogether they can be classified into 4 categories. First category is flavin-based electron carriers or flower proteins that represent the lowest affinity of electron carriers and are present within complex 1 and 2. Second class are cytochromes, the proteins which are could be subclassified in 3 categories: cytochromes A, B, and C, and generally has high electron affinities as compared to the um, flavin-based electron carriers. The iron sulfur cluster electron carriers are the largest and most abundant class within electron transport chain and are spread around, present within various components of the respiratory chain, have a vast variety of the affinities for electrons. And the last fourth standalone class, which could be defined as ubiquinone, which we have already discussed.